Hi everyone, my name is Erica Almance, and my current research is about water uptake strategies in urban green landscapes, specifically the Dallas-Fort Worth Metroplex. And I've been working with Dr. Ricardo Sanchez Murillo. So we know that urban green infrastructure is any system that serves to mitigate water pollution and floods from flashy stormwater episodes. However, there's little understanding of the role that plants play in partitioning the urban water cycle. So what water sources do different plant species prefer? Well, we used naturally occurring tracers, or in our case, water isotopes, to examine how precipitation inputs are stored in the soil and taken up by plants. So to collect precipitation, we used a Palmex rain collector, which has a bottle in here from which we get the rainwater. We also collected soil water from suction lysimeters at depths of 5, 15, and 30 inches. And monthly, we collected all of the above, as well as some stem samples and discrete soil samples. These stem samples were from 3 different tree species that are common to the DFW area: the cherry laurel, the elderberry, and box elder maple. These are the photos of those, and we sampled the same exact tree each time. And of course, we sampled some surface water as well. That just involves filling a 30 milliliter sample bottle with some of the surface water, whether it be from the swamp or the stream. And the water from those stem and soil samples were extracted via centrifugation at 11,000 RPM for 1.5 hours. And lastly, all the water samples were analyzed using our isotope analyzer, which uses laser spectroscopy. So this tells us things like the delta O18 or oxygen 18 composition, the delta deuterium composition, deuterium excess, and even delta O17. So for our results, with that data, we can see a few things. So here we have a plot of the delta O18 of our data. We can see that water extracted via centrifugation and suction lysimeters exhibited similar isotope compositions versus soil depth, and this water also agreed with recent precipitation in the DFW area, shaded here in red and orange. The surface water compositions resulted in more enriched values, and the source water here corresponds to an inlet from Lake Worth. And we can also see in this graph that there are the 3 tree species shown here in the blue. So we have the cherry laurel, the box elder maple, and the elderberry. And the red part again shows the isotopic composition of the precipitation. So based on the plot, the water from the cherry laurel and the elderberry trees right here both have compositions that agree with that of the precipitation. And this suggests that they uptake rainwater fairly quickly and they prefer to use recent precipitation. The box elder maple tree, however, right here, exhibited more enriched values, and this suggests that it has an older water residence time. And here we have our Bayesian isotope mixing model. So this mixing model, overall, the plots here show that mixing contributions indicated that homogeneous soil moisture from recent precipitation was the main water source for the trees. But for the box elder maple, shows that over half the contribution was from the stream. And there was no differences by soil depth reported. The significance of this project is quite interesting. This information can assist in urban planning, because if a team is trying to design an urban green landscape, studies like this one can help them select trees depending on their water uptake strategies. For example, trees like cherry laurel that uptake recent precipitation might be better options for flood mitigation. Further research is definitely needed in this area to advance our understanding and application of urban green infrastructure for water quality management in highly altered and fast-growing urban areas like our own. We would like to thank the Fort Worth Botanic Gardens, the UT System STARS program, and the Office of the Provost for their support of this project. And thank you to all of you for watching.